Every quiet moment inside the body is busier than it seems, even when you are sitting still, breathing softly and doing nothing more dramatic than existing. The nervous system is awake in the background. It is listening to the room through your ears, noticing pressure through your skin, keeping track of where your arms and legs are, adjusting your heartbeat, guiding your breath, and quietly helping your thoughts move from one idea to the next. It is the hidden network that lets the outside world become experience. The nervous system is often described as electrical, and that is partly true. Tiny signals move through nerve cells with remarkable speed, carrying information from the senses to the brain, from the brain to the muscles, and from the organs back into the deeper control centers of the body. But it is not simply a bundle of wires. It is also chemical, flexible, protective, and astonishingly selective. It does not just pass messages along. It filters them, reshapes them, compares them with memory, and decides what should rise into awareness and what can remain quietly unnoticed. This is why a soft sound can pull your attention across a room, why a familiar smell can bring back a memory, why your hand moves away from something hot before you have fully thought about it, and why the body can keep breathing through the night without needing a polite reminder every few seconds. The nervous system is not only the machinery of thought, it is the machinery of sensation, movement, balance, sleep, emotion, survival, and the strange private feeling of being here at all. So tonight, the aim is to slow all of that down, not because the nervous system itself is slow, because it absolutely is not. It is more like a very excitable messenger service that has had too much coffee, except far more organized and much less likely to misplace a parcel. But by moving gently through it, piece by piece, the hidden structure becomes easier to see. Welcome to the late lab. It's lovely to have you here. Before we go any further, where in the world are you watching from tonight? Are you tucked into bed after a long day, sitting somewhere quiet with the lights low, resting on a sofa while the room slowly forgets to be busy? Wherever you are, I hope you feel comfortable, safe, and ready to let the evening soften around you. If you enjoy calm, thoughtful journeys through science and would like more of them drifting your way, a like and subscribe really does help this sleepy little channel continue. And if you're already subscribed, thank you, truly. Your support means a great deal, and it helps keep these quiet explorations alive. Tonight's subject is the nervous system, which does come with a small warning. It may be a little too interesting for sleep if you start thinking too hard about the fact that your own brain is currently using itself to understand itself. That is a slightly unfair trick from biology. But the journey will stay gentle, steady, and clear. So let's begin slowly. What is the nervous system, and how does this hidden web of cells turn tiny signals into sensation, movement, memory, emotion, breath, balance, and the quiet feeling of being alive? You're awake in a room, perhaps more still than you have been all day, and yet stillness is only the surface of the story. There is sound arriving at your ears, even if it is only the soft hush of the room around you. There is pressure where your body meets the chair, the bed, or the floor. There is a quiet sense of where your hands are, even without looking at them. Your lungs are moving. Your heart is beating. Your eyes are gathering light. Your mind is following words, turning sounds into meaning, and gently placing that meaning somewhere inside the wider landscape of thought. It can feel simple, almost effortless, as if being awake is just something that happens. But beneath that ordinary experience, the nervous system is working with extraordinary speed and delicacy. It is sending signals, receiving signals, filtering signals, and deciding which signals deserve attention and which can be allowed to fade into the background. It is the body's fast communication network, linking sensation, movement, memory, emotion, breathing, balance, digestion, and awareness into one coordinated living system. If the body's an orchestra, the nervous system is not only the conductor, it is also part of the sheet music, several of the musicians, and the person in the back making absolutely sure nobody comes in early with the cymbals. The nervous system allows the body to respond quickly because it uses specialized cells called neurons. Neurons are the main signaling cells of the nervous system. They're shaped for communication, often with branching structures that receive information and long fibers that carry messages across distance. Some neurons connect across tiny spaces inside the brain. Others send fibers from the spinal cord all the way towards muscles in the hands, feet, or face. They do not shout across the body in words, of course. They communicate through tiny changes in electrical charge, through chemical messengers, through membranes that open and close channels, and through ions moving in carefully controlled patterns. Ions are charged particles such as sodium, potassium, calcium, and chloride. These tiny particles matter because nerve cells use their movement to create electrical signals. A neuron maintains a difference in charge between the inside and outside of its membrane. When the right conditions are met, that balance shifts suddenly, creating a travelling electrical impulse called an action potential. This impulse can move along the neuron's axon, carrying information with impressive precision. It is not electricity in quite the same way as electricity moving through a household wire, but the comparison is understandable. The nervous system does use electrical differences. It simply does so in a wet, living, chemical, beautifully fussy biological way. Neurons do not work alone. They are supported by glial cells, which were once treated as little more than nervous system packing material. That was a little unfair, rather like calling an entire theatre crew some people near the curtains. Glial cells help maintain the environment around neurons, provide insulation, support repair, guide development, assist immune defense, and help keep signaling stable. Some glial cells, called oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system, produce myelin. Myelin is a fatty insulating layer around many nerve fibers. It allows signals to travel much faster and more efficiently, a little like adding proper insulation to a communication cable, except the cable is alive and occasionally involved in deciding whether you noticed a draft. The nervous system is usually divided into two broad parts. The central nervous system consists of the brain and spinal cord. This is the main processing and command region, where enormous amounts of information are received, compared, interpreted, and coordinated. The brain is protected by the skull, and the spinal cord runs down through the vertebral column, carrying signals between the brain and the rest of the body. The peripheral nervous system includes the branching nerves that extend outward from the brain and spinal cord, reaching the skin, muscles, joints, organs, and blood vessels. These nerves form the living roots through which the central nervous system stays in conversation with the body. That conversation is constant. A sensory nerve in the skin may report pressure, temperature, vibration, or pain. A motor nerve may carry a command to a muscle so the body can move. Other nerves help adjust the heart, lungs, stomach, intestines, glands, and blood vessels, often without asking for conscious permission. This is why breathing can continue while attention drifts. Digestion can proceed while thoughts wander elsewhere, and balance can be adjusted before a person has time to write a formal complaint to gravity. Yet the nervous system is not a simple wire network. That image is useful for a moment, then it quickly becomes too small. A wire carries a signal from one place to another. The nervous system does far more. It filters information before awareness receives it. It predicts what might happen next. It adjusts sensitivity depending on context. It suppresses signals that are not important. It interprets patterns, compares them with memory, and builds a practical version of reality that the body can use. The sensation of sitting quietly in a room is not a raw recording of the room. It is an organized experience, shaped by the brain and body together. This is one reason the nervous system can feel so mysterious. It is both physical and personal. Its work is built from cells, membranes, ions, chemical transmitters, and electrical impulses, yet the result is the feeling of being present. A tiny change in nerve signaling can mean the touch of fabric, the sound of rain, the urge to blink, the memory of a face, or the sudden realization that the foot has gone slightly numb because it has been tucked under the leg for too long. The body can be poetic when it wants to be, but it is also very willing to send practical complaints. The scale of the nervous system is astonishing. The human brain contains roughly 86 billion neurons, and these neurons form connections through synapses, the tiny communication points between cells. Across the brain, there are often estimated to be hundreds of trillions of synaptic connections. These numbers are not meant to make the mind feel overcrowded, although it does explain why thinking about dinner can somehow become thinking about childhood, a song lyric, and whether the oven was left on. They show that nervous system activity is deeply networked. A thought, sensation, or movement is not usually the work of one cell. It emerges from patterns across many cells acting together. The scientific understanding of neurons develops slowly. In the late 19th century, Camille Golgi created a staining method that revealed individual nerve cells with remarkable clarity. His technique coloured only some cells, but those cells appeared in striking detail, like dark trees against a pale background. Santiago Ramon y Cajal used Golgi's method to study nerve tissue and argued that the nervous system was made of separate cells rather than one continuous web. This became known as the neuron doctrine. It was a major shift in neuroscience because it showed that the nervous system's communication depends on individual cells meeting and signalling across tiny gaps rather than one unbroken mesh. Golgi and Ramon y Cajal shared the Nobel Prize in Physiology or Medicine in 1906, although they disagreed strongly about how the nervous system was organized. It is one of science's more elegant little awkward moments. Two researchers honored together while not fully agreeing about what the evidence meant. Over time, the neuron doctrine became central to modern neuroscience. It gave scientists a clearer framework for understanding how nerve cells communicate, how circuits form, and how the living tissue of the brain and body can produce coordinated behavior. Even now, the nervous system remains one of the most complex structures known in biology. Signals can move at different speeds depending on the type of nerve fiber and whether it is myelinated. Some slow pain signals travel at only a few meters per second, while heavily myelinated fibers can carry signals far faster, over 100 meters per second in some cases. Speed matters, but timing matters just as much. The nervous system is not merely trying to send messages quickly. It is trying to send the right message to the right place at the right moment with the right level of urgency. So the quiet electricity inside you is not just a spark or a current. It is a living language. It is chemical, electrical, cellular, and deeply organized. It allows the body to notice the world, move through it, remember it, respond to it, and remain balanced inside it. Every soft breath, every faint sound, every passing thought, every small adjustment of posture depends on this hidden network, working in darkness, without applause, and usually without being noticed at all. And perhaps that is the most remarkable part. The nervous system does not need to announce itself to shape every moment of experience. It works quietly beneath awareness, turning charged particles and chemical signals into the ordinary miracle of being awake, alive, and gently connected to the world around you. To understand the nervous system, it helps to move from the whole shimmering network down to the individual living cells that make it possible. The brain, spinal cord, and branching nerves may seem like large structures, but their work depends on tiny messengers, each one shaped by biology to receive information, process it, and pass it along. These cells are called neurons, and they are among the most specialized cells in the body. A neuron is not a simple wire. It may look wire-like in diagrams, especially when shown with a long thin fibre stretching away from a central body, but it is alive in every important sense. It uses energy. It repairs itself where it can. It builds proteins, maintains membranes, moves materials internally, responds to chemical signals, and constantly adjusts its own sensitivity. A neuron is closer to a tiny living decision point than a passive cable. It is also rather fussy, which is fair enough, considering that the wrong signal at the wrong time can be the difference between a graceful movement and accidentally walking into a doorframe like the doorframe was personally invited to the conversation. Most neurons have several basic parts. The dendrites are the branching structures that receive incoming signals from other neurons. Their name comes from a word meaning tree, and that is a helpful image, because dendrites often spread like delicate branches from the cell body. These branches are covered with receptors, tiny molecular structures that respond to chemical messengers released by neighboring cells. A dendrite is not just a passive receiver. It helps shape the incoming information, deciding how strongly a signal affects the neuron and how that signal combines with many others arriving at nearly the same time. The cell body, also called the soma, contains the nucleus and much of the machinery that keeps the neuron alive. Inside it, genes are read, proteins are made, energy is managed, and the cell's basic maintenance is carried out. Mitochondria supply usable energy, which neurons need in generous amounts because signaling is expensive work. Membranes must be maintained. Ion gradients must be restored. Transport systems must move molecules and small structures along the neuron's length. For a cell that often looks elegantly simple in a textbook drawing, a neuron is running a surprisingly busy internal logistics department. From the cell body extends the axon, a long projection specialized for carrying signals away from the neuron. Some axons are extremely short, connecting nearby cells within a small local circuit. Others are much longer, especially in motor neurons that send fibers from the spinal cord toward muscles in the limbs. The axon begins at a region called the axon hillock, where incoming signals are effectively gathered and tested. If the combined activity is strong enough, the neuron reaches threshold and an electrical impulse begins. At the far end of the axon are axon terminals. These terminals form connections with other cells at synapses, the tiny meeting points where one neuron influences another neuron, a muscle cell or a gland cell. At many synapses, the electrical impulse arriving at the terminal causes chemical messengers called neurotransmitters to be released. These neurotransmitters cross a microscopic gap and bind to receptors on the next cell. In that moment, electricity becomes chemistry, and chemistry may become electricity again. The nervous system is very fond of changing languages mid-sentence, but somehow it usually remains wonderfully coherent. One of the most important features of a neuron is its resting membrane potential. This means that even when a neuron is not actively firing, there is a small electrical difference between the inside and outside of its membrane. The inside of the neuron is usually more negatively charged than the outside. This difference exists because ions such as sodium, potassium, chloride, and calcium are distributed unevenly across the membrane, and because the membrane contains channels and pumps that control how those ions move. A useful way to imagine this is to picture the neuron as quietly prepared. It is not asleep. It is ready. The membrane is maintaining a delicate imbalance, like a drawn bow or a calm lake held behind a gate. The sodium-potassium pump helps maintain this state by moving sodium ions out of the neuron and potassium ions in. Ion channels then allow certain ions to move under specific conditions. The result is not random electrical noise, but controlled biological readiness. When a neuron receives enough input to reach threshold, voltage-gated ion channels open in the membrane. Sodium ions rush inward, changing the voltage across the membrane. Then potassium channels help restore the balance as potassium ions move outward. This rapid travelling change in membrane voltage is called an action potential. It moves along the axon like a wave of electrical change, not because the same bit of electricity flows all the way from one end to the other, but because each section of membrane triggers the next section. It is a travelling event, a carefully timed ripple of living electricity. The modern understanding of this process owes a great deal to Alan Hodgkin and Andrew Huxley. In the mid-20th century, they studied the squid giant axon, which was large enough to make detailed electrical measurements easier than in many other nerve fibers. Their work showed how changes in sodium and potassium conductance could explain the action potential. In other words, they helped reveal how nerve impulses emerge from the controlled movement of ions through the neuron's membrane. The squid, perhaps unknowingly, made a very generous contribution to neuroscience. It did not fill in the paperwork, but science appreciated it all the same. Action potentials are often described as all-or-nothing events. Once the threshold is reached, the impulse fires in a consistent way. But that does not mean all nerve signals carry the same meaning. The nervous system does not encode information by making one action potential for happiness, another for pain, and another for remembering where the biscuits are kept. Meaning depends on timing, firing frequency, connection patterns, receptor types, and the wider circuit receiving the signal. A rapid burst of impulses can mean something different from a slow rhythm. A signal arriving in one pathway can produce a different experience or action from the same kind of electrical event arriving somewhere else. Different neurons are specialized with different roles. Sensory neurons carry information from the body and the outside world toward the central nervous system. They help detect light, sound, pressure, temperature, pain, stretch, and chemical signals. Motor neurons carry commands from the central nervous system toward muscles, allowing movement, posture, facial expression, swallowing, and many other actions. Interneurons sit between neurons within the brain and spinal cord, shaping local circuits, refining signals, and helping create the complex patterns behind thought and behavior. In the cerebral cortex, pyramidal neurons are especially important for long-range communication between brain regions and for many forms of perception, planning, and voluntary movement. They have a distinctive pyramid-shaped cell body and long dendrites that help them gather input from many sources. In the cerebellum, Purkinje cells are large, highly branched neurons involved in coordination, timing, balance, and motor learning. Their dendritic trees can look almost impossibly delicate, like living lace arranged for computation. In the spinal cord, motor neurons send commands outward to muscles, translating nervous system activity into movement that can be seen, felt, and measured. The variety matters because the nervous system is not built from identical units doing identical jobs. It is built from specialized cells arranged into circuits. Some neurons carry information over long distances. Some refine signals locally. Some excite their neighbors. Some inhibit them. Some respond to a specific kind of sensory input. Others integrate many kinds of information at once. The shape, chemistry, location, and connections of a neuron all influence what it does. So the neuron is both simple enough to name and complex enough to spend a lifetime studying. It is a living messenger, but also a processor, a receiver, a responder, and a participant in vast networks. It depends on membranes, ions, mitochondria, proteins, receptors, transport systems, and repair processes. Its signals are electrical, chemical, rhythmic, and contextual. And from countless neurons communicating together, the nervous system begins to produce the larger patterns of sensation, movement, memory, emotion, and awareness. The quiet electricity inside the body is therefore not floating freely through empty space. It is carried by living cells, each one maintaining its own delicate conditions, listening to its neighbours, and passing along messages when the moment is right. Beneath every thought and every movement, neurons are doing this patient work, turning tiny shifts in charge into the language of life. The electrical impulse travelling along a neuron is only part of the story. It moves down the axon as a tiny wave of changing voltage passing through living membrane. But sooner or later that impulse reaches the end of the neuron. It arrives at the axon terminal, and there it faces a small but important problem. The next cell is usually not physically joined to it. There is a gap. That gap is called a synapse. More precisely, in many cases it is a chemical synapse, a tiny space where one neuron can influence another without touching it directly. The sending neuron is called the presynaptic neuron. The receiving cell is called the postsynaptic cell. Between them is the synaptic cleft, a gap so small it is measured in nanometers, yet important enough that thought, movement, sensation, memory, and emotion all depend on what happens there. A great deal of being alive takes place in spaces too small to see without powerful instruments. When an action potential reaches the axon terminal, it changes the voltage of the terminal membrane. This opens voltage-gated calcium channels, allowing calcium ions to enter the terminal. Calcium helps trigger tiny membrane-bound packages called synaptic vesicles to move towards the cell membrane and fuse with it. These vesicles contain chemical messengers called neurotransmitters. Once released, neurotransmitters drift across the synaptic cleft and bind to receptors on the postsynaptic cell. This is the handover at the heart of neural communication. Electrical activity becomes chemical release, and chemical binding may then become electrical activity again. The nervous system is constantly translating between forms of signalling. It is not simply firing sparks through wires. It is opening channels, releasing molecules, binding receptors, clearing chemicals away, and adjusting the strength of its own connections. Neurotransmitters are often spoken about as if each one has a simple personality. Dopamine becomes pleasure, serotonin becomes happiness, endorphins become comfort. This is memorable, but far too neat. In reality, neurotransmitters do different things depending on where they're released, which receptors they bind to, and what circuit they're working within. Dopamine is involved in motivation, reward prediction, movement, learning, and attention. Serotonin contributes to mood, sleep, appetite, pain processing, and many other functions. Norepinephrine helps regulate arousal, alertness, stress responses, and attention. Endorphins are involved in pain modulation and reward, but they are not simply the body's tiny cheerleaders, waving little biochemical flags. Glutamate is the main excitatory neurotransmitter in the brain, meaning it often makes postsynaptic neurons more likely to fire. Gamma-aminobutyric acid is the main inhibitory neurotransmitter in the brain, meaning it often makes neurons less likely to fire. This balance between excitation and inhibition is essential. If excitation dominates too strongly, circuits can become unstable. If inhibition dominates too strongly, signaling may be suppressed. The nervous system depends on controlled tension between activity and restraint. In a way, it is the biological version of a calm conversation where everyone gets to speak, but nobody is allowed to bring a trumpet. Acetylcholine is another important neurotransmitter. It acts at the neuromuscular junction, where motor neurons communicate with skeletal muscle fibers, allowing movement. It also plays roles in attention, learning, memory, and autonomic nervous system function. The discovery that nerves could communicate chemically was one of the great turning points in neuroscience. Otto Loewi studied frog hearts and showed that stimulating the vagus nerve released a substance that could slow the heartbeat. This substance was later identified as acetylcholine. Henry Dale also made major contributions to understanding chemical transmission and acetylcholine's role in the nervous system. Together, Loewi and Dale helped establish that nerves use chemical messengers. Chemical synapses must clear their signals after release. If neurotransmitters simply remained in the synaptic cleft, messages would blur together and circuits would lose precision. Some neurotransmitters are taken back into the presynaptic neuron through reuptake transporters. Some are broken down by enzymes. Some diffuse away and are absorbed or processed by nearby cells, including glial cells. Acetylcholine, for example, is broken down by the enzyme acetylcholinesterase. This clearing process allows synapses to reset, making communication sharp enough for repeated signaling. Receptors shape what happens after a neurotransmitter arrives. Ionotropic receptors are fast. When a neurotransmitter binds, the receptor opens an ion channel directly, allowing charged particles to move across the membrane. This can quickly change the electrical state of the postsynaptic cell. Metabotropic receptors are slower and more indirect. They activate internal signaling pathways inside the cell, often involving G proteins and second messenger systems. These effects may take longer to begin, but they can last longer and change how responsive the cell becomes. One type is like opening a door immediately. The other is more like sending a message to the building manager, who then adjusts the lighting. There are also electrical synapses, though they are less common in the human nervous system than chemical synapses. In electrical synapses, cells are connected by gap junctions, allowing ions to pass directly between them. This can make signaling extremely fast and can help groups of cells act together. Chemical synapses, however, allow more flexibility. They can amplify signals, weaken signals, reverse effects through different receptors, and change with experience. This flexibility is one reason synapses matter so deeply for learning and memory. Synapses are not fixed little switches. Their strength can change. A connection that is used repeatedly may become more effective. Another may weaken over time. This is part of synaptic plasticity, the nervous system's ability to alter communication between cells. The brain is continually adjusting through real biological changes in receptors, vesicle release, gene activity, and circuit organization. So thoughts, movements, sensations, and emotions do not come from one magic chemical or one isolated signal. They arise from patterns across many neurons, many synapses, and many receptor types. A single neurotransmitter can participate in different experiences depending on the circuit. A single synapse can influence whether a neuron fires now, later, more easily, or not at all. The meaning of a signal is created by timing, location, chemistry, and context. At every moment, synapses are quietly shaping the nervous system's conversation with itself. They are the places where cells pause without quite touching, where electricity becomes chemistry, where chemistry changes living membrane, and where the private world of thought begins to emerge from countless tiny exchanges. Inside those microscopic gaps, the nervous system does not merely pass messages along. It edits them, weights them, clears them, strengthens them, and sometimes lets them fade, giving the brain and body the flexibility to keep learning from the world. The nervous system is often introduced through neurons, and that makes sense. Neurons carry signals, form circuits, and create the flashing pattern of communication that lets the body sense, move, think, and respond. But neurons are not floating through empty space, heroically doing everything alone like tiny overworked messengers with no lunch break. Around them is another vast family of cells, quieter in reputation, but absolutely essential to the nervous system's survival. These are glial cells. The word glia comes from an old idea that these cells were a kind of glue holding the nervous system together. In the 19th century, the German pathologist Rudolf Virchow introduced the term neuroglia, meaning nerve glue. At the time, that seemed reasonable. Under early microscopes, these cells did not appear to fire obvious electrical impulses like neurons, so they were easy to underestimate. But modern neuroscience has changed that view completely. Glial cells are not passive packing material. They support, protect, insulate, nourish, regulate, repair, and sometimes reshape the conditions that allow neurons to communicate at all. A neuron may carry the dramatic signal, but glial cells help make the stage safe enough for that signal to happen. Without them, the nervous system would be chemically unstable, poorly insulated, vulnerable to damage, and much less able to maintain the delicate environment that electrical and chemical signaling require. They're a little like the backstage crew of the brain, rarely the ones taking the bow, but if they vanished, the whole performance would rapidly become confusing, dangerous, and badly lit. One important type of glial cell is the astrocyte. Astrocytes are named for their star-like shape, with many branching arms reaching around neurons, synapses, and blood vessels. They help maintain the chemical environment around neurons by regulating ions and neurotransmitters outside cells. This matters because neurons depend on precise conditions. If potassium levels rise too much around neurons, or if neurotransmitters linger where they should not, signaling can become distorted. Astrocytes help keep the neighborhood tidy. Astrocytes also support synapses, the tiny communication points between neurons. They can take up neurotransmitters such as glutamate, help recycle chemical ingredients, and influence how synapses function over time. This means that communication between neurons is not only a two-cell conversation. In many cases, astrocytes are nearby, listening in and helping regulate the exchange. They are not gossiping exactly, but they are definitely aware of the local drama. Astrocytes also help regulate blood flow in the brain. Active neurons need oxygen and glucose, and changes in neural activity can be linked to changes in local blood supply. This relationship is one of the reasons brain imaging methods can detect activity indirectly. Astrocytes are also involved in the blood-brain barrier, a protective system formed mainly by specialized blood vessel cells, supported by astrocyte end feet and other cellular structures. The blood-brain barrier helps control what can pass from the bloodstream into brain tissue, protecting the brain from many harmful substances while allowing essential materials to enter. Another major glial role is insulation, and this brings in oligodendrocytes and Schwann cells. Both produce myelin, a fatty insulating layer that wraps around many axons. Oligodendrocytes form myelin in the central nervous system, meaning the brain and spinal cord. Schwann cells form myelin in the peripheral nervous system, meaning the nerves that branch out through the body. Myelin does not cover every axon, but where it is present, it can make signaling much faster and more efficient. Myelin works by allowing action potentials to travel in a special way called saltatory conduction. Rather than regenerating the electrical impulse along every tiny patch of membrane, the signal effectively jumps between exposed gaps in the myelin. These gaps are called nodes of Ranvier. The action potential is renewed at each node, allowing the impulse to move rapidly along the axon. It is not quite teleportation, although the nervous system would probably enjoy the branding. It is a clever biological shortcut, using insulation and exposed nodes to speed communication while saving energy. This is why myelin is so important for coordinated movement, quick reflexes, sensory precision, and efficient communication between distant parts of the nervous system. In some myelinated fibers, signals can travel at well over 100 meters per second. In slower unmyelinated fibers, signals may move at only a few meters per second or less. The nervous system uses both fast and slow signaling depending on the job. Sharp touch, position sense, and rapid motor commands benefit from speed. Duller pain signals and certain internal body signals may move more slowly. When myelin is damaged, nervous system communication can become disrupted. Multiple sclerosis is one important example. It is an immune-mediated condition in which myelin in the central nervous system is damaged, producing problems that can vary widely depending on which pathways are affected. Symptoms may involve vision, movement, sensation, balance, fatigue, or coordination. The point is not that myelin is merely useful. It is that reliable signaling depends on this insulation, and when it is lost, messages can slow, distort, or fail. Microglia are another essential type of glial cell. They are immune-related cells within the central nervous system, constantly surveying their surroundings. When there is injury, infection, inflammation, or cellular debris, microglia can respond. They help clear damaged material and participate in immune defense. They are sometimes described as the resident immune cells of the brain and spinal cord, although their role is more nuanced than simply acting like tiny security guards with clipboards. Microglia are also involved in synaptic pruning, especially during development. Synaptic pruning is the process by which some connections between neurons are removed or refined. This helps shape nervous system circuits as the brain develops and adapts. It may sound strange that removing connections can be useful, but a healthy nervous system is not built by keeping every possible link forever. It needs organization, selectivity, and refinement. A brain with no pruning would not be more advanced. It would be more like a room where nobody ever throws away old cables. Glial cells therefore show that nervous system signaling depends on much more than the moment when one neuron talks to another. Synapses need stable chemistry. Axons need support and insulation. Brain tissue needs protection, nourishment, blood flow regulation, immune surveillance, and careful cleanup. The nervous system is not only a network of messages, it is an ecosystem of living cells maintaining the conditions that make those messages meaningful. So while neurons often receive the attention, glial cells are the quiet support system beneath the whole conversation. They do not usually produce the obvious action potentials that make neurons famous, but they shape the world in which those action potentials can happen. They help the nervous system stay balanced, protected, efficient, and adaptable. In the soft darkness of the brain and the long branching nerves of the body, glial cells are always there, keeping the living circuitry steady enough for thought, sensation, movement, and memory to continue. By the time nerve signals have crossed synapses, moved through glial-supported tissue, and passed from one cell to another, they begin to form something larger than individual messages. They become patterns. Those patterns rise and settle across the brain, a soft, folded, energy-hungry organ that sits protected inside the skull, quietly turning biological signals into movement, memory, emotion, attention, perception, and the feeling of being awake inside a body. The brain is often called a command center, and that is useful as long as it is not taken too literally. It does help control the body, but it is not a little person sitting behind a desk, stamping forms and giving orders to the kidneys. It is more like a living control landscape, constantly receiving information, comparing it with memory, adjusting body systems, predicting what might happen next, and shaping behavior before awareness has even had time to clear its throat. Although the brain is only a small fraction of body weight, it uses a large share of the body's resting energy. Much of that energy is not spent on dramatic thoughts or sudden flashes of genius. It is spent maintaining the basic conditions that neurons need in order to signal, restoring ion gradients, recycling neurotransmitters, supporting cell membranes, regulating synapses, and keeping billions of cells alive and responsive. The brain is expensive tissue. It is soft, delicate, and metabolically demanding, which is a rather bold combination for something so important. The outer surface of the brain is dominated by the cerebral cortex. This is the folded outer layer involved in perception, voluntary movement, language, planning, memory, attention, and flexible behavior. Its folds increase surface area, allowing more cortical tissue to fit inside the skull. These folds are not decorative, although they do give the brain the appearance of a walnut that has spent several million years becoming unusually thoughtful. Different parts of the cerebral cortex are associated with different functions. The primary visual cortex, at the back of the brain in the occipital lobe, is crucial for early processing of visual information. The primary motor cortex near the top and side of the brain in the frontal lobe helps control voluntary movement. The somatosensory cortex receives information from the body, including touch, pressure, and body position. The prefrontal cortex supports planning, decision-making, attention, impulse control, and the flexible organization of behavior. But the brain is not divided into neat little boxes where one region does one job alone. Most mental and bodily functions depend on distributed networks. Seeing, speaking, remembering, moving, feeling afraid, solving a problem, or recognizing a familiar face all involve cooperation across many regions. A specialized area may be very important, but it is almost never working in isolation. The brain is less like a row of separate offices and more like a city at night, with many districts lighting up in changing patterns depending on what is happening. Some specialized regions became famous because injury revealed their importance. In the 19th century, Paul Broca studied patients with damage in part of the left frontal lobe who had difficulty producing speech, even though they could often understand language. This region became known as Broca's area. Carl Wernicke later described patients with damage farther back in the left hemisphere who could produce fluent speech that often lacked meaning, while also struggling to understand spoken language. This region became known as Wernicke's area. Their work helped show that language depends on identifiable brain systems, while later research has shown that real language processing is broader and more networked than those two areas alone. Beneath the cortex are deeper structures that help organize the brain's constant activity. The thalamus is a major relay and filtering center, especially for sensory information traveling toward the cortex. It helps regulate what reaches conscious processing, although it is not merely a passive relay station. The hypothalamus is small but deeply important, helping regulate hunger, thirst, body temperature, sleep rhythms, hormone control, and autonomic functions. It is one of those brain regions that proves size is not everything, a lesson the hypothalamus seems quietly smug about. The hippocampus, tucked deep within the temporal lobe, is essential for forming many new memories and for linking experience with place and context. The amygdala nearby is involved in detecting emotional significance, learning associations, and shaping responses to threat or importance. It is often simplified as the brain's fear center, but that label is too narrow. The amygdala is better understood as part of a wider system that helps the brain decide what matters. The basal ganglia are a group of deep brain structures involved in movement, habit, action selection, reward learning, and the smooth initiation or suppression of behavior. They help the brain decide not only how to move, but which action is worth allowing through. The cerebellum, sitting at the back and lower part of the brain, is famous for balance and coordination, but it also supports timing, motor learning, and aspects of cognition. It contains a huge number of neurons and helps make movement accurate rather than wobbly, delayed, and suspiciously puppet-like. The brainstem connects the brain with the spinal cord and supports some of the most basic functions of life, including breathing rhythms, heart rate regulation, swallowing, arousal, and sleep-wake control. It is not glamorous in the way the cortex is often made to seem, but it is absolutely essential. A brilliant thought is lovely, but breathing is generally considered helpful first. The corpus callosum is the thick bundle of nerve fibers connecting the left and right cerebral hemispheres. It allows information to pass between the two sides, helping them work together. The hemispheres do show some specialization, especially in language and spatial processing, but the popular idea of one side being purely logical and the other purely creative is far too simple. Both hemispheres are deeply involved in complex human thought. Scientists study the living brain using several major tools. Magnetic resonance imaging shows detailed brain structure. Functional magnetic resonance imaging measures changes related to blood oxygenation, giving an indirect view of brain activity. Positron emission tomography can track metabolism or specific chemical processes using radioactive tracers. Electroencephalography records electrical activity from the scalp, offering excellent timing even though it gives less precise location. Each method reveals part of the story, like looking at the same quiet city through maps, lights, traffic patterns, and distant weather. The brain, then, is not a single master switch. It is an organ of coordination. Its billions of neurons and even larger numbers of synapses create shifting patterns that control movement, interpret sensation, regulate the body, support memory, generate emotion, and help construct conscious experience. From tiny ion movements and chemical signals, whole body behavior begins to emerge. The soft command center is not commanding from a distance. It is woven into everything the body feels, does, remembers, and quietly becomes. The brain may feel like the center of the nervous system story, but it does not sit alone inside the skull, quietly issuing instructions into empty space. Its signals need a route into the body. Sensory information needs a route back up. Between the brain and the wider landscape of muscles, skin, joints and organs, runs one of the most important structures in the body, the spinal cord. The spinal cord is the main communication pathway between the brain and the body. It carries sensory information upward towards the brain, including touch, pressure, vibration, pain, temperature, and body position. It also carries motor commands downward from the brain towards muscles, allowing movement, posture, balance, and fine control. It is easy to imagine it as a cable, but that image is only partly useful. The spinal cord is not a passive bundle of wires. It is living nervous tissue, capable of organizing reflexes, shaping signals, and helping coordinate movement before conscious awareness has fully arrived on the scene with a clipboard. The spinal cord runs down from the brainstem through the vertebral column, protected by the bones of the spine. Around it are protective membranes called meninges, the same broad family of membranes that help protect the brain. These layers surround the central nervous system and help cushion and support delicate tissue. The spinal cord is also bathed in cerebrospinal fluid, which provides further protection and helps maintain a stable environment. For something so central to movement and sensation, the spinal cord is treated by the body with appropriate caution, like a very important cable that is also somehow alive, opinionated, and easily offended by poor posture. Inside the spinal cord there is grey matter and white matter. The grey matter sits more centrally and contains many neuron cell bodies, interneurons, synapses, and local circuits. In cross-section, it is often described as butterfly-shaped, although it is perhaps one of the least whimsical butterflies in biology. Around it lies white matter, made mostly of myelinated nerve fibers carrying signals up and down the cord. The white colour comes from myelin, the fatty insulating layer that helps many nerve signals travel quickly and efficiently. Signals enter and leave the spinal cord through roots. The dorsal roots carry sensory information into the spinal cord from the body. The ventral roots carry motor commands out from the spinal cord towards muscles. These roots join to form spinal nerves, which branch outward through the body. This organization gives the spinal cord a beautifully practical design. Information from the body enters through one root, commands to the body leave through another, and local circuits can process some signals without needing to send every small decision all the way to the brain first. This becomes especially clear in reflexes. A reflex is a rapid, automatic response to a stimulus. The knee-jerk reflex is a familiar example. When a tendon below the kneecap is tapped, stretch receptors in the muscle detect a sudden change. Sensory neurons carry that information into the spinal cord. There, the signal connects with motor neurons that send a command back to the muscle, causing the lower leg to kick forward slightly. Conscious awareness may notice the movement, but the basic response does not need to wait for a thoughtful committee meeting in the cerebral cortex. The withdrawal reflex is another useful example. If the hand touches something painfully hot, sensory neurons carry warning signals into the spinal cord. Local circuits can activate motor neurons that pull the hand away before the brain has fully processed the details. The brain is still informed, of course, which is why the experience becomes painful and memorable. But the first movement can begin closer to the source of danger. This is not the body being rude to the brain. It is the body being efficient. In emergencies, politeness is less important than not leaving a finger on a hot pan. The British neurophysiologist Charles Sherrington was central to understanding reflexes and the integrated action of the nervous system. He helped develop the concept of the synapse, studied how reflexes are coordinated, and showed that the nervous system works as an organized whole rather than a loose collection of isolated reactions. His work helped reveal that movement and response depend on integration, excitation, inhibition, timing, sensory feedback, and motor output all woven together. The spinal cord also contains major ascending and descending tracts. Ascending tracts carry information upward towards the brain. The dorsal column pathway carries fine touch, vibration, and conscious body position sense. This is part of how the brain knows where the limbs are without needing to look at them every second, which is useful, because constantly checking where the elbows have gone would make daily life unnecessarily theatrical. The spinothalamic tract carries pain, temperature, and crude touch information upward. It helps the brain become aware of signals that may require protection, attention, or adjustment. Descending tracts carry commands downward from the brain. The corticospinal tract is especially important for voluntary movement, particularly skilled control of the limbs and hands. It begins in areas of the cerebral cortex involved in movement and travels down through the brainstem and spinal cord, influencing motor neurons and interneurons. This pathway helps turn intention into action, allowing a thought such as reaching for a cup to become a carefully shaped movement of shoulder, arm, wrist, hand, and fingers. Because these pathways are organized along the length of the spinal cord, injuries can have very different effects depending on where they occur and how severe they are. Damage higher in the spinal cord can affect more of the body than damage lower down, because more pathways have not yet branched out to their targets. Damage to sensory pathways may affect sensation. Damage to motor pathways may affect movement. Damage may be complete or incomplete, and the details matter enormously. The spinal cord is highly organized, so the pattern of disruption depends on which regions and pathways are involved. Yet the spinal cord is not only important when something goes wrong, it is active in ordinary life every second. It helps maintain posture when standing. It contributes to walking patterns through spinal circuits that coordinate rhythmic movement. It carries the feeling of the floor under the feet and the position of joints in space. It helps the body respond to touch, pressure, discomfort, and stretch. It is part highway, part processing center, part reflex organizer, and part quiet negotiator between the brain and the body. This makes the spinal cord one of the nervous system's most elegant compromises. It extends the central nervous system downward into the body, keeping the brain connected to distant tissues, while also allowing some responses to be handled close to where they are needed. It carries messages, but it also shapes them. It supports conscious movement, but it also manages automatic reactions. It is hidden inside the spine, rarely noticed when everything is working well, yet every step, stretch, shiver, and small adjustment of balance depends on its steady communication. The spinal cord is therefore not just a pathway beneath the brain. It is a living information highway with local intelligence built into its lanes. Through grey matter, white matter, dorsal roots, ventral roots, spinal nerves, reflex arcs, and long tracts, it keeps the body in conversation with itself. And beneath the calm surface of ordinary posture and movement, signals are rising and falling through it constantly, linking the soft command center in the skull with the moving, sensing body below. The spinal cord carries the nervous system's traffic downward into the body, but it does not reach every fingertip, toe, muscle, gland, and organ by itself. From that central pathway, nerves branch outward, dividing again and again until they form a vast living network outside the brain and spinal cord. This is the peripheral nervous system. It is the part of the nervous system that reaches into the skin, muscles, joints, organs, glands, and blood vessels, keeping the central nervous system in constant contact with the rest of the body. The word peripheral can make it sound secondary, as if these nerves are somewhere around the edges doing the less glamorous paperwork. But without the peripheral nervous system, the brain and spinal cord would be sealed away from the body they're supposed to guide. Sensation would not arrive properly, movement commands would not reach muscles, internal organs would lose important lines of communication. The central nervous system may be protected inside bone, but the peripheral nerves are the branching roots that let its signals touch the living world. Peripheral nerves can be grouped in several useful ways. Cranial nerves emerge directly from the brain or brainstem. There are 12 pairs of cranial nerves, and they help control or sense parts of the head, neck, and some internal organs. They're involved in smell, vision, eye movement, facial expression, hearing, balance, swallowing, taste, and more. One of the most important cranial nerves is the vagus nerve. The vagus nerve travels from the brainstem down into the chest and abdomen, carrying information between the brain and internal organs, including the heart, lungs, and digestive tract. Its name comes from a word meaning wandering, which is fair because it takes a rather ambitious route through the body. Spinal nerves, by contrast, emerge from the spinal cord. They branch out through spaces between the vertebrae and spread into the body. These nerves carry sensory information inward and motor information outward. They help the skin report touch, pressure, pain, temperature, and vibration. They help muscles receive instructions for movement. They carry information from joints and muscles about body position, helping the nervous system know where the limbs are even when the eyes are closed. This sense is called proprioception, and without it, ordinary movement would become strangely uncertain, as if the body had misplaced its own map. Within these nerves are different kinds of fibers. Sensory fibers carry information from the body toward the central nervous system. A cool surface beneath the hand, pressure under the foot, a stretch in the shoulder, or discomfort in a joint can all be carried inward through sensory pathways. Motor fibers carry commands from the central nervous system toward muscles and glands. When a hand reaches for a cup, when posture adjusts slightly, or when a facial muscle moves into a small expression, motor fibers are part of the pathway that makes the action possible. Some of this activity belongs to the somatic nervous system. The somatic nervous system is involved in voluntary movement and conscious sensation. It helps control skeletal muscles and carries much of the sensory information that can rise into awareness. When you choose to move a hand, shift position, or notice the texture of fabric, the somatic nervous system is involved. Yet even here, not everything becomes conscious. The body is constantly sending information upward, and much of it is filtered, adjusted, or used quietly in the background. Awareness receives the edited version, not the full inbox. This is probably merciful. Nobody needs a live report from every square centimetre of skin at once. A peripheral nerve is not just one exposed thread. It is made of many axons bundled together, wrapped and supported by layers of connective tissue. These layers help protect the fibers and organize them into a durable structure. Peripheral nerves also need a blood supply, because nerve cells and their long fibers require oxygen, nutrients, and stable conditions. Many peripheral axons are wrapped in myelin made by Schwann cells. This myelin helps signals travel more quickly and efficiently, just as myelin made by oligodendrocytes supports many axons in the brain and spinal cord. Schwann cells do more than insulate. In the peripheral nervous system, they can help guide repair after certain kinds of nerve injury, although recovery depends on many factors, including the type, location, and severity of damage. This is one important difference between many peripheral nerves and the central nervous system, where repair is often more limited. Even so, peripheral nerves are delicate living structures, not simple wires that can be unplugged and replaced like a charger cable found at the bottom of a drawer. Doctors and researchers can study peripheral nerve and muscle function using tests such as nerve conduction studies and electromyography. Nerve conduction studies measure how quickly and effectively electrical signals travel along a nerve. Electromyography records electrical activity in muscles, often helping to show whether a problem may involve muscle, nerve, or the connection between them. These tools do not tell the entire story of a person's nervous system, but they can provide useful information about how signals are moving through peripheral pathways. The peripheral nervous system is easiest to appreciate through ordinary moments. Touch a cool glass, and sensory fibers report temperature and pressure. Stand up, and nerves from muscles and joints help the central nervous system adjust balance and posture. Feel the ground beneath one foot, and pressure receptors send information inward. Decide to move a hand, and motor fibers carry the command outward. Meanwhile, many other signals continue below awareness, including information from internal organs, blood vessels, and glands. So the peripheral nerves are not merely extensions of the spinal cord. They are the nervous system's reach into the body. They carry the central nervous system's traffic into muscles and organs, and they bring the body's news back inward. Through cranial nerves, spinal nerves, sensory fibers, motor fibers, Schwann cells, connective tissue, and countless branching roots, the body remains connected to itself. Every touch, adjustment, breath, swallow, step, and quiet shift of posture depends on this hidden network, spreading through the body like fine roots through living ground. The peripheral nerves reach through the body like branching roots, but what they carry is not one simple kind of message. They carry many forms of sensation, each translated from the physical world into the language of the nervous system. A cool surface, a bright room, a rising sound, a bitter taste, a stretched muscle, a sharp pain, or the quiet awareness of where the feet are in space all begin as different kinds of physical events. Somehow, the nervous system has to turn those events into electrical activity. That process is called sensory transduction. It is the conversion of physical energy, chemical presence, temperature change, pressure, movement, or tissue damage into signals that sensory neurons can carry. Light is not itself a nerve impulse. Sound is not itself a nerve impulse. Pressure on the skin is not itself a nerve impulse. Each must be detected by specialized receptor cells or sensory nerve endings, then translated into changes in membrane voltage. The world arrives in many forms, but the nervous system has to speak about it using cells, ions, receptors, and carefully timed electrical signals. Different senses begin with different kinds of receptors. Mechanoreceptors respond to physical force, such as touch, pressure, vibration, stretch, and movement. They help the skin detect texture, the inner ear detect sound and balance, and muscles and joints report position. Thermoreceptors respond to temperature. Nociceptors respond to potentially damaging stimuli, such as extreme heat, strong pressure, inflammation, or chemical irritation. Photoreceptors in the retina respond to light. Chemoreceptors respond to chemicals, allowing taste and smell, and also helping the body monitor internal conditions. Proprioceptors report the position and movement of muscles, tendons, and joints, giving the body its quiet sense of itself. This means sensation is not one doorway into the body. It is a whole set of doorways, each tuned to something different. Touch, pressure, vibration, temperature, pain, proprioception, hearing, balance, taste, smell, and vision all give the nervous system different kinds of information. The brain does not simply receive a single stream called the outside world. It receives many streams, each travelling along particular pathways, carrying information that has already been filtered and shaped before it reaches awareness. In the 19th century, the German physiologist Johannes Müller described an important idea known as the doctrine of specific nerve energies. The basic point was that the brain interprets a signal partly according to which sensory pathway is active, not just according to the raw physical cause. If the visual pathway is activated, the experience is visual. If an auditory pathway is activated, the experience is sound. This helps explain why very different events can sometimes produce similar kinds of sensation if they affect the same pathway. Pressing gently on a closed eye, for example, can create little visual flashes, not because light has entered, but because the visual system has been mechanically disturbed. The brain reads the route as part of the message. Vision begins when photoreceptors in the retina respond to light. Rod cells are especially useful in dim conditions, while cone cells support color vision and fine detail. Hearing begins when sound waves move structures in the ear, eventually bending tiny hair cells in the cochlea. Balance also depends on hair cells, but in the vestibular system, where fluid movement and tiny structures help detect head motion and orientation. Taste uses chemoreceptors on the tongue and elsewhere in the mouth, while smell depends on olfactory receptors high in the nasal cavity, where airborne molecules can influence sensory neurons directly. Touch can feel simple, but it is made of several channels. Some receptors detect light touch, others detect deep pressure, stretch, or vibration. Proprioception is even quieter. It allows the body to know where its parts are without constant visual checking. You can close your eyes and still touch a finger to your nose, not because the finger sent a postcard, but because muscles, tendons, joints, and sensory pathways are continually reporting position and movement. This sense is easy to ignore until it is disrupted, at which point the body can suddenly feel strangely uncertain. Pain is more complicated still. It begins with nociception, the detection of potentially harmful stimuli by nociceptors, But pain is not just a raw alarm signal travelling upward unchanged. It is a protective sensory and emotional experience shaped by spinal processing, brain interpretation, inflammation, attention, expectation, memory, and context. A small injury may feel worse when someone is tired, frightened, or focused intensely on it. A more serious injury may briefly feel less intense during danger or distraction. This does not mean pain is imaginary. It means pain is actively constructed by the nervous system as a protective experience, influenced by what the body and brain believe matters in that moment. Ronald Melzack and Patrick Wall introduced the gate control theory of pain in the 20th century. Their theory helped explain how pain signals can be modulated in the spinal cord before they reach full conscious experience. Activity in some nerve fibers can influence whether pain-related signals are amplified or dampened. This is one reason rubbing a bumped elbow may reduce discomfort for a while. Touch-related input can affect spinal processing, partly closing the gate on pain signals. The theory has been refined over time, but it remains an important landmark in understanding pain as something shaped by the nervous system, not merely passed along like a message in a tube. The brain also brings expectation and attention into perception. It does not passively record the world like a camera left running on a table. It constructs a usable picture from incoming signals, prior experience, body state, and the present situation. The brain predicts what is likely, checks sensory information against those predictions, and updates when necessary. This is usually helpful. It allows perception to be quick, efficient, and stable. It also explains why the nervous system can sometimes be fooled by illusions, missing details, or mistaken expectations. The brain is not trying to be deceptive. It is trying to be useful, which is not always exactly the same thing. So sensation is not the world entering the body untouched. It is the world being translated. Pressure becomes patterns of nerve activity. Light becomes signals in the retina. Sound becomes movement, then receptor activity, then electrical messages. Chemical traces become taste or smell. Tissue threat becomes nociception and sometimes pain. Body position becomes proprioception. Every sense begins in physical contact with reality, but perception emerges only after the nervous system has interpreted the signals. Through sensory transduction, specialized receptors, peripheral nerves, spinal pathways, and brain networks, the body builds a living picture of itself and its surroundings. It is not a perfect picture, and it is not a simple recording. It is practical, flexible, and constantly updated. It lets the body know where it is, what it is touching, what may be dangerous, what may be useful, and what deserves attention. In every quiet moment, the world is entering the nervous system not as sound, light, heat, or pressure exactly, but as the electrical language of living cells softly shaped into experience. Sensation brings the world into the nervous system, but movement sends the body back out into the world. A touch, a sound, a change in balance, or a thought can all lead to action. The hand reaches, the eyes shift, the shoulders settle, the foot adjusts against the floor. Movement can feel simple because it happens so smoothly. But behind every voluntary action is a long chain of planning, signaling, feedback, and correction. Voluntary movement often begins with intention. That intention is not a single spark in one tiny corner of the brain. It emerges from activity across networks involved in attention, memory, motivation, body position, and decision-making. Before a hand moves towards a cup, the nervous system has already considered where the cup is, where the arm is, how much force may be needed, what posture will support the movement, and how to keep the rest of the body from joining in too enthusiastically. Reaching for, T should not require the legs to panic. Several brain regions help prepare and guide movement. The primary motor cortex sends many of the signals involved in voluntary control, especially skilled movement of the hands, face, and limbs. The premotor cortex helps plan actions in relation to the outside world, such as reaching towards an object. The supplementary motor area is involved in internally generated movement sequences, especially actions that unfold in an organized order. These regions do not work alone. They cooperate with deeper and lower structures, including the basal ganglia, cerebellum, brainstem motor centers, spinal cord circuits, and spinal motor neurons. One famous way of visualizing movement control is the motor homunculus. This is a map of body representation in the primary motor cortex, developed through cortical mapping studies. The hands, lips, tongue, and face take up large areas because they require fine control. The result is often drawn as a strange little figure with enormous hands and lips, which is useful for teaching but should not be mistaken for a tiny person living in the brain. The brain does not contain a miniature manager pulling levers. It contains networks of neurons whose activity corresponds to control over different parts of the body. The Canadian neurosurgeon Wilder Penfield helped make cortical mapping famous through work with patients undergoing brain surgery. By gently stimulating parts of the exposed cortex while patients were awake under local anesthesia, Penfield and his colleagues could observe sensations or movements linked to different regions. This work contributed to modern understanding of how the cortex represents the body, though it sits within a much wider history of neuroscience, neurology, and careful clinical research. The maps were revealing, but they were never the whole story. Once a movement command leaves the brain, it travels through descending pathways, including the corticospinal tract. This pathway carries signals from the cerebral cortex down through the brainstem and spinal cord. In the spinal cord, these signals influence motor neurons directly or through interneurons. The motor neurons then send impulses out through peripheral nerves towards muscles. At this point, the nervous system's intention is becoming physical action. The final meeting point between nerve and muscle is the neuromuscular junction. Here, a motor neuron communicates with a skeletal muscle fiber. When an action potential reaches the motor neuron's terminal, acetylcholine is released into the small gap between nerve and muscle. Acetylcholine binds to receptors on the muscle fiber, triggering electrical changes in the muscle membrane. This leads to calcium release inside the muscle fiber, allowing the protein machinery of contraction to work. In simple terms, nerve signaling gives the muscle permission to contract, and the muscle responds by pulling. But movement is not just command followed by obedience. The nervous system constantly compares intended motion with actual body position. Sensory feedback from muscles, tendons, joints, skin, and the inner ear tells the brain and spinal cord what is really happening. If the arm is slightly off course, corrections can be made. If the ground is uneven, posture changes. If an object is heavier than expected, grip force increases. Movement is a conversation between prediction and feedback, not a one-way announcement from the brain to the body. The cerebellum is especially important for this kind of refinement. It helps with timing, coordination, balance, motor learning, and error correction. It receives information about intended movements and actual movements, then helps adjust future commands so actions become smoother and more accurate. Without proper cerebellar function, movements may become clumsy, poorly timed, or unstable. The cerebellum helps prevent the body from moving like a badly animated puppet discovering gravity for the first time, which is generally appreciated by everyone nearby. The basal ganglia play a different but equally important role. They are involved in movement selection, habit, initiation, and suppression. They help decide which actions should be allowed through and which should be held back. This is crucial because the nervous system is always capable of many possible movements. but only some are useful at any given moment. The basal ganglia help turn intention into chosen action, while preventing unwanted movements from spilling out. Parkinson's disease shows how deeply movement depends on these circuits. In Parkinson's disease, dopamine-producing neurons in a region called the substantia nigra are gradually lost. This disrupts basal ganglia function and can lead to symptoms such as slowed movement, stiffness, tremor, and difficulty initiating action. The condition is not simply a muscle problem. It is a disorder of motor circuits, chemical signaling, timing, and action control. It reveals how movement depends on the careful cooperation of brain regions that usually work quietly beneath awareness. Even the simplest action is therefore beautifully layered. A person decides to move, but that decision depends on perception, memory, motivation, and body state. Brain regions prepare the action. Spinal pathways carry commands. Peripheral nerves deliver signals. The neuromuscular junction turns chemical communication into muscle activation. Sensory feedback checks the result. The cerebellum refines timing and accuracy. The basal ganglia help select and release the right action. Movement feels natural because the nervous system hides most of the work. It allows intention to become action without requiring awareness of every pathway, synapse, muscle fiber, and correction. The hand moves, the body balances, the foot steps, and the head turns towards a sound. Beneath that smoothness, countless signals are being shaped, compared, and adjusted so the body can move through the world with quiet precision. Voluntary movement makes the nervous system feel active and outward-facing. A thought becomes a reaching hand. A shift in balance becomes a careful step. A decision becomes movement through the world. But even when the body seems still, the nervous system is not resting in the way a switched-off machine rests. It is regulating the inside of the body, quietly adjusting organs, blood vessels, glands, digestion, heart rate, breathing patterns, temperature, and pupil size. Much of this work belongs to the autonomic nervous system. The autonomic nervous system helps control functions that usually do not require conscious command. You do not normally have to instruct the heart to beat faster when climbing stairs, or ask the pupils to widen in a dark room, or remind the intestines to keep moving food along at a civilized pace. The body manages these tasks through automatic regulation. Automatic does not mean simple. It means the control is usually handled below awareness, through networks that keep the internal body stable and responsive. The autonomic nervous system is often divided into the sympathetic nervous system, the parasympathetic nervous system, and the enteric nervous system. These systems are sometimes presented as neat opposites, with the sympathetic system as the accelerator and the parasympathetic system as the brake. That image can be helpful for a moment, but it is too simple. In real life, they interact, overlap, and shift the body's internal state depending on need. The body is not a car with two pedals. It is more like a living weather system, constantly changing pressure, flow, heat, and rhythm. The sympathetic nervous system helps prepare the body for increased demand. During sympathetic activation, alertness may rise, heart rate may increase, blood pressure may shift, pupils may dilate, and glucose may be mobilized so tissues have more available fuel. Blood flow can be redirected, with more support for skeletal muscles, and less emphasis on digestion in moments of urgency. Sweating may increase. Breathing patterns may change. This is often connected with the phrase fight or flight, a concept associated with the American physiologist Walter Cannon. Cannon also helped develop the idea of homeostasis, the body's ability to maintain internal stability despite changing conditions. Homeostasis is important because the autonomic nervous system is not only for danger. It helps the body adjust through ordinary life. Standing up, eating a meal, entering a cold room, hearing a sudden noise, feeling embarrassed, exercising gently, or settling down to sleep all involve changes in internal regulation. The sympathetic system is not simply a panic button. It is part of a flexible control network that helps the body meet changing demands, from serious stress to the deeply dramatic event of realizing someone is watching while you try to parallel park. The parasympathetic nervous system is often associated with rest, digestion, and recovery. It helps slow the heart rate, supports digestive activity, contributes to gland secretion, and allows the body to shift towards maintenance and restoration. A major pathway of parasympathetic communication is the vagus nerve. The vagus nerve travels from the brainstem into the chest and abdomen, carrying signals between the brain and organs such as the heart, lungs, and digestive tract. It is one of the most important communication routes between the brain and the internal body. Parasympathetic activity does not simply turn the body off, it helps regulate. It can slow the heart, support digestion, influence breathing patterns, and help the body settle after demand has passed. When the body is resting after a meal, parasympathetic pathways help make digestion possible. When the heart needs to slow after exertion, parasympathetic influence helps bring it back towards a calmer rhythm. It is not laziness, it is maintenance. And maintenance is one of biology's least glamorous miracles. The enteric nervous system is another essential part of the picture. It is sometimes called the nervous system of the gut because it contains large networks of neurons embedded in the walls of the digestive tract. These neurons help coordinate muscle contractions, secretions, blood flow, and local digestive activity. The enteric nervous system can manage many digestive processes locally, although it also communicates with the brain through autonomic pathways, including the vagus nerve. This gut-brain communication helps link digestion with appetite, nausea, stress, fullness, and internal comfort. Stress research was shaped by Hans Selye, who studied how the body responds to prolonged demand. His work helped popularize the idea that stress involves whole-body adaptation, not just a brief emotional reaction. Modern science has refined and expanded those ideas, but the broad lesson remains useful. The nervous system, hormones, immune activity, metabolism, and behavior all interact when the body is under pressure. Stress is not just a feeling in the mind, it is a coordinated biological state. The autonomic nervous system also contributes to interoception, the sensing of the internal body. Interoception includes signals related to heartbeat, breath, hunger, fullness, nausea, warmth, discomfort, thirst, and internal tension. These signals help the brain monitor the state of the body from within. Some interoceptive information becomes conscious, such as noticing a racing heart or an empty stomach. Much of it stays below awareness, quietly guiding regulation and behavior. The body is always reporting on itself, though thankfully it does not send all its updates as notifications. This hidden regulation shows that the nervous system is just as busy in stillness as it is in movement. While a hand rests, the heart is being adjusted. While the eyes grow heavy, breathing is being shaped. While digestion continues, blood flow, temperature, glands, and internal organs are being regulated. The autonomic nervous system keeps the inner world flexible enough to meet the outer one, shifting the body gently between alertness, activity, recovery, digestion, and rest. So the body on autopilot is not unattended. It is carefully watched from within by living circuits that sense, respond, and adjust. Through sympathetic activation, parasympathetic regulation, enteric coordination, interoception, and homeostasis, the nervous system keeps the internal body in quiet conversation with itself. Even in a calm room, even in the softest stillness, the body is being guided moment by moment, not by conscious effort, but by an ancient automatic intelligence working beneath awareness. The autonomic nervous system shows that the body is constantly regulating itself from within. Heart rate, breathing, digestion, blood pressure, temperature, and internal comfort are all being adjusted quietly below awareness. But these internal changes are not separate from emotion. They are part of it. An emotion is not just a thought floating in the mind or a feeling labelled neatly after it appears. It is a whole body nervous system process involving perception, memory, attention, hormones, facial expression, action readiness, and the changing state of the body itself. A sudden sound in the dark is a useful place to begin. Before there is a clear thought, the body may already respond. The heart may beat faster, breathing may shift, muscles may tighten, attention sharpens, the eyes may widen, digestion may pause its quiet work, presumably offended but understanding. This is not because emotion lives in one little compartment of the brain. It is because the nervous system rapidly links sensory information with memory, prediction, bodily preparation, and conscious feeling. Several brain regions are important in emotion and threat processing. The amygdala helps detect emotional significance and supports learning about important or threatening events. The hypothalamus links the brain with autonomic and hormonal responses, helping adjust internal body state. The prefrontal cortex helps with interpretation, planning, impulse control, and emotional regulation. The insula is strongly involved in sensing the internal body, including heartbeat, nausea, breath, warmth, and discomfort. The anterior cingulate cortex helps connect emotion, attention, pain, decision-making, and action. The hippocampus gives context through memory, helping the brain distinguish between a real present danger and something that merely resembles one. The brainstem supports arousal, facial expression, breathing changes, and basic survival responses. The amygdala is often called the fear center, but that phrase is too small. The amygdala is not a tiny alarm bell that only knows how to panic. It is involved in detecting significance, forming associations, and shaping responses to emotionally important stimuli. It helps the nervous system learn that a certain sound, smell, face, place, or situation might matter. Sometimes that significance is threatening. Sometimes it is rewarding, surprising, socially important, or linked to memory. Fear is part of the story, but not the whole story. The neuroscientist Joseph Ledoux is well known for his work on fear circuits and the pathways by which sensory information can influence emotional responses. His research helped clarify how threat-related processing can involve routes through the thalamus, amygdala, and cortex, allowing the nervous system to respond quickly while more detailed interpretation continues. Modern emotion science, however, is broader than any single pathway. Emotion involves networks across the brain and body, not one simple line from stimulus to feeling. When the nervous system detects a serious challenge, the brain can also communicate with the endocrine system. One major pathway is called the hypothalamic pituitary adrenal axis. In simple terms, the hypothalamus signals the pituitary gland, and the pituitary gland signals the adrenal glands which sit above the kidneys. The adrenal glands can then release hormones including cortisol. Cortisol helps mobilize energy, influence immune activity, support alertness, and help the body respond to prolonged demand. This is slower than a reflex or a sudden nerve impulse, but it can shape the body's state for minutes or longer. This stress response is not automatically bad. It is part of how the body adapts. The problem comes when stress is intense, repeated, or prolonged without enough recovery. Then systems that are useful in short bursts can become wearing. The nervous system is built for change, not for being held in the same high alert setting indefinitely, like a smoke alarm that has discovered toast and decided to make it everyone's problem. Emotion is deeply tied to bodily state. A racing heart can contribute to the feeling of fear or excitement. Slow breathing can help shift the body towards calm. Muscle tension can make the world feel more threatening. A settled stomach can make a situation easier to tolerate. Digestion, posture, facial expression, body temperature, pain, and fatigue all influence emotional experience. The body is not just receiving emotions from the brain, it is helping build them. Facial expression is part of this loop as well. Emotional states can shape the muscles of the face, and facial feedback may also influence how feelings are experienced. A smile, a grimace, widened eyes, or a tightened jaw can all become part of the nervous system's wider emotional pattern. None of this means emotions are fake or easily controlled by simply changing posture. It means feelings are embodied. They happen through a relationship between brain, body, environment, and interpretation. Attention also matters. The nervous system tends to give priority to information that seems relevant to safety, reward, social connection, or uncertainty. If something feels threatening, attention may narrow around it. If something feels safe and familiar, the body may allow attention to soften. Memory joins in too. A place, sound, smell, or facial expression can gain emotional meaning because of past experience. The hippocampus helps provide context, while the amygdala helps attach significance. Together, they allow the nervous system to ask not only what is happening, but whether something like this has mattered before. Emotional regulation is often misunderstood. It does not mean suppressing feelings or pretending nothing is happening. It means adjusting attention, interpretation, body state, and behavior so that emotion can be understood and guided rather than simply obeyed. The prefrontal cortex is important here because it helps evaluate situations, reframe meaning, inhibit impulsive reactions, and choose responses. The limbic system, including the amygdala, hippocampus, and related regions, contributes emotional significance and memory. Regulation emerges through communication between these networks, along with signals from the body. This is why calming the body can sometimes help calm the mind. Slow breathing, gentle movement, warmth, social safety, and reduced sensory threat can all influence autonomic state. Equally, changing interpretation can influence the body. Realizing that a loud noise was only a door closing can reduce alarm. Remembering that a racing heart came from climbing stairs, not danger, can shift the feeling attached to it. The nervous system keeps updating the meaning of the body's signals. Emotion, then, is not a weakness in the system. It is part of how the nervous system prepares the body for action, connection, protection, learning, and adaptation. Fear, joy, anger, sadness, disgust, surprise, calm, curiosity, and affection all involve patterns across circuits, hormones, muscles, memories, and internal sensations. They're not just thoughts, they're not just chemicals, they are living states of the whole organism. The body's alarms can be loud, but they are not random. They are attempts to protect, prioritize, and prepare. Sometimes they are accurate. Sometimes they are shaped by old learning, tiredness, pain, or expectation. But always they show how closely internal regulation and feeling are intertwined. Beneath every emotional moment, the nervous system is listening to the world, listening to the body, comparing the present with memory, and deciding what kind of state may be needed next. Emotion gives the nervous system a way of deciding what matters, but memory lets those meanings last. A sound can become familiar. A face can become important. A place can feel safe or uneasy because the nervous system has linked it with past experience. Memory is not a single storage box tucked neatly inside the brain, like a little filing cabinet with suspiciously good handwriting. It is a set of processes spread across cells, circuits, and time. Memory begins with encoding. This is the process by which the nervous system takes an experience and begins turning it into a form that can be stored. Attention matters here. Emotional significance matters too. Something surprising, meaningful, frightening, beautiful, painful, or repeated is more likely to be encoded than something the brain quietly judges as background noise. This is one reason a person may remember an awkward comment from years ago but forget why they walked into the kitchen 30 seconds ago. The nervous system is powerful, but it has priorities, and sometimes those priorities are unhelpfully dramatic. After encoding comes consolidation. Consolidation is the gradual stabilizing of memory over time. Some memories remain fragile at first, then become more durable through changes in synapses, circuits, and broader brain networks. Retrieval is the process of bringing a memory back into use. But retrieval is not like opening a sealed recording. Remembering can change the memory. This is called reconsolidation, where a recalled memory may become temporarily flexible before being stored again. Forgetting is also part of the system, not merely a failure. The brain must reduce, blur, or lose some information. Otherwise, every detail of every day would pile up like an endless cupboard full of unlabeled cables. Interference happens when memories overlap or compete, making recall harder. One of the most important structures for forming many new declarative memories is the hippocampus. Declarative memories are memories that can usually be consciously described, such as facts and events. The hippocampus sits deep in the temporal lobe and helps bind together different elements of experience, where something happened, when it happened, what was seen, what was heard, and what it meant. It is especially important for forming new episodic memories, the memories of lived events. But long-term memory is not stored only in the hippocampus. Over time, many memories depend on distributed networks across the cerebral cortex. This became clearer through the famous case of Henry Molaison, often known for many years by his initials in scientific writing. In 1953, he underwent surgery to treat severe epilepsy, and parts of his medial temporal lobes, including much of the hippocampus, were removed. Afterward, he could still hold information briefly in mind and remember many things from before the surgery, but he was profoundly impaired in forming new long-term declarative memories. The neuropsychologist Brenda Milner studied him carefully, and her work helped reveal the crucial role of the hippocampus in memory formation. His case also showed that memory is not one single ability, because some forms of learning remained possible even when new conscious memory was deeply disrupted. At the cellular level, learning depends partly on synaptic plasticity. Synaptic plasticity means that the strength of communication between neurons can change. One important form is long-term potentiation, where repeated activity can make a synapse more effective. Another is long-term depression, where synaptic strength can decrease. These processes help neural circuits adapt through experience. They do not explain all of memory by themselves, but they are central to understanding how the nervous system changes when it learns. The psychologist Donald Hebb helped shape modern thinking about learning with what is often called the Hebbian learning principle. The broad idea is that when neurons are active together in a meaningful pattern, the connections between them can become stronger. This is sometimes summarized informally as cells that fire together wire together, though in real biology the details are more complicated. The useful point is that the nervous system is changed by patterned activity. Experience leaves traces by altering the probability that certain circuits will activate together again. Not all memory is conscious memory. Procedural memory involves skills and habits, such as riding a bicycle, typing, playing an instrument, or tying shoelaces. These memories rely heavily on circuits involving the basal ganglia and cerebellum. With repetition, actions can become smoother, faster, and less dependent on conscious attention. At first, a new skill may feel clumsy and effortful. Over time, the nervous system refines timing, force, sequence, and prediction. Eventually, the body may seem to know what to do before the mind has finished narrating it. Sleep also matters for learning and memory. During slow-wave sleep, the brain shows large coordinated patterns of activity, and research suggests that memories can be replayed and reorganized. The hippocampus appears to replay patterns linked to recent experience, helping train wider cortical networks. Rapid eye movement sleep is associated with vivid dreaming and may support aspects of emotional memory, creativity, and synaptic regulation, although sleep science is still refining the details. Across the night, the brain does not simply switch off. It sorts, strengthens, weakens, and integrates information. Sleep is not lost time. It is more like the nervous system doing careful housekeeping, while consciousness is politely asked to stop interfering. Emotion and memory are closely linked, because emotional significance helps decide what the nervous system prioritizes. The amygdala can influence memory systems when something seems important, especially during threat, reward, or strong feeling. Stress hormones can also affect memory, sometimes strengthening recall of central details while making surrounding details less clear. This is why emotionally charged memories can feel vivid, but not always perfectly accurate. A strong feeling is not the same as a perfect recording. Memory is therefore both powerful and imperfect. It lets the nervous system learn from the past, but it also reshapes the past each time it is brought into the present. It helps the body avoid danger, repeat useful actions, recognize people, build skills, understand language, and maintain a sense of self through time. From changing synapses to sleeping brain rhythms, from the hippocampus to the cortex, from conscious recollection to quiet habit, memory shows that the nervous system is not fixed. It is always being rewritten gently and biologically by the life it has already lived. Memory lets the nervous system learn from the past, but attention decides what part of the present receives priority. At every moment, the brain is surrounded by far more information than it can treat equally. Light is entering the eyes, sound is reaching the ears, the skin is reporting pressure, temperature, and texture. Muscles and joints are quietly announcing body position. Internal signals from the heart, lungs, stomach, and blood vessels are also arriving. If all of it became equally loud in awareness, ordinary life would feel less like consciousness and more like being trapped inside an over-enthusiastic control room. Attention is the nervous system's way of selecting, amplifying, suppressing, and prioritizing information. It helps decide what becomes clear, what stays faint, and what disappears into the background. This does not mean ignored information vanishes completely. Much of it is still processed enough to guide behavior. A person can be focused on reading and still adjust posture, notice a sudden sound, or look up when their name is spoken nearby. Attention is not a single spotlight shining from one place in the brain. It is a shifting set of processes that changes how strongly different signals influence perception, thought, and action. Selective attention is the ability to focus on one stream of information while reducing the influence of others. A classic everyday example is hearing your name in a noisy room. The sound may have been present among many voices, but once it becomes relevant, the nervous system lifts it out of the blur. Sustained attention is the ability to remain focused over time, such as following a long explanation, or watching the road during a quiet drive. Divided attention is the attempt to handle more than one task at once, although the brain is not always as good at this as confidence suggests. Executive control helps manage attention deliberately, allowing a person to resist distraction, switch tasks, hold goals in mind, and choose what matters now. One important structure in attention is the thalamus. The thalamus sits deep in the brain and acts as a major relay and filtering center, especially for sensory information traveling towards the cerebral cortex. It is sometimes described as a relay station, but that makes it sound too passive. The thalamus helps regulate the flow of information, shaping what reaches cortical processing and how strongly it arrives. It is involved in alertness, sensory selection, sleep, consciousness, and the coordination of activity across brain networks. Attention also depends on broad networks across the cortex, brainstem, basal ganglia, and frontal regions. The brainstem contributes to arousal and wakefulness, helping set the general level of alertness. The basal ganglia help with selection, not only of movements, but also of actions and cognitive priorities. Frontal regions, especially parts of the prefrontal cortex, help guide attention according to goals, plans, and rules. Parietal regions help direct attention through space, allowing the nervous system to focus on where something is happening as well as what it is. The brain is also constantly predicting. Predictive processing is a modern theoretical framework in neuroscience that suggests the brain uses prior expectations to interpret incoming sensory information. It does not wait passively for the world to explain itself. It makes best guesses, compares those guesses with incoming signals, and updates when there is a mismatch. This can make perception faster and more efficient. It is one reason a familiar room can be understood almost instantly, while an unusual shadow or unexpected sound catches attention. Carl Friston is strongly associated with the free energy principle and predictive processing. His work is mathematically complex, but the gentle version is that the brain and body try to reduce uncertainty by improving predictions about sensory input and acting in ways that keep the organism within workable conditions. In plain language, the nervous system is always asking what is likely to be happening, what should be noticed, and whether the current signals fit the expected pattern. It is less like a camera and more like a careful interpreter, though one that occasionally makes confident mistakes before breakfast. This is why perception can be useful and accurate enough to guide survival while still being actively constructed. The brain does not create reality from nothing, but it also does not simply record reality without interpretation. It builds a practical model from incoming signals, memory, attention, body state, and expectation. Most of the time, this works extremely well. It lets the body move through the world smoothly, recognize objects, understand speech, avoid danger, and find the doorway instead of politely walking into the wall. But because perception is constructed, it can be fooled by illusions, assumptions, fatigue, fear, or missing information. Large-scale brain networks help organize these shifting states. The default mode network is often active during inward-focused thought, such as remembering, imagining, mind-wandering, and thinking about the self. The frontoparietal control network helps with task control, flexible thinking, planning, and deliberate attention. The salience network helps detect relevant changes, especially events that may matter for action, emotion, or bodily state. These networks do not act like separate departments with locked doors. They interact, compete, and cooperate as the brain moves between rest, focus, reflection, and response. Past experience shapes all of this. Memory teaches the nervous system what patterns are familiar, what deserves caution, what is usually safe, and what has mattered before. A sound in one setting may be ignored, while the same sound in another setting may sharpen attention. A smell may barely register until it is linked with a memory. A face may stand out in a crowd because it is familiar. Attention and prediction are therefore built on learning. The brain uses the past not as a perfect guide, but as a starting point for understanding the present. So the brain's inner filter is not a barrier between the person and the world. It is the reason the world becomes manageable. Attention gives priority. Prediction gives context. Memory gives meaning. Together, they allow the nervous system to turn a flood of signals into an organized experience, clear enough to act on, flexible enough to update, and quiet enough that the mind can rest inside it without hearing every signal at once. Attention is the nervous system's way of choosing what matters while the body is awake. Sleep changes that relationship. The outside world does not disappear, but the nervous system turns down many of its usual channels, reshapes awareness, and begins doing work that is difficult to see from the outside. A sleeping person may look still, but the brain is not simply switched off. Sleep is an active nervous system state, with changing brain rhythms, shifting body regulation, memory processing, sensory disconnection, and quiet cellular maintenance. Sleep is usually divided into non-rapid eye movement sleep and rapid eye movement sleep. Non-rapid eye movement sleep includes lighter stages and deeper slow-wave sleep. During slow-wave sleep, large populations of neurons in the cortex move through slower, more synchronized patterns of activity. The body becomes less responsive to the outside world. Breathing and heart rate often settle, and the brain enters a state strongly linked with physical restoration and memory consolidation. It is not unconsciousness in the ordinary sense of a machine going dark. It is more like the nervous system changing its operating mode. Rapid eye movement sleep is different. During rapid eye movement sleep, brain activity becomes more wake-like in some ways, and vivid dreaming is more common. The eyes move beneath closed lids, breathing and heart rate may become more variable, and most skeletal muscles are placed into a temporary state of paralysis called muscle atonia. This prevents the body from acting out dreams in full, which is generally helpful. Without it, a dream about running through a forest could become an unfortunate disagreement with the bedside table. The discovery of rapid eye movement sleep is strongly linked to Nathaniel Kleitman and Eugene Aserinsky. In the early 1950s, Aserinsky, working with Kleitman, observed periods of rapid eye movements during sleep and connected them with dreaming. Later, Michel Jouvet made major contributions to understanding rapid eye movement sleep physiology, including the role of brainstem mechanisms and muscle atonia. Their work helped show that sleep is not one uniform state, but a repeating cycle of distinct nervous system patterns. Sleep is shaped by two major forces. One is the circadian rhythm, the body's roughly daily timing system. A tiny structure in the hypothalamus called the suprachiasmatic nucleus helps coordinate this rhythm, using light information from the eyes to align the body with the day-night cycle. The hormone melatonin, released by the pineal gland, helps signal biological night, although it is not a simple sleep switch. It is more like a soft message that evening has arrived. The other force is sleep pressure. The longer a person stays awake, the more the need for sleep builds. One chemical involved in this process is adenosine, which accumulates during waking and contributes to sleepiness. Caffeine works partly by blocking adenosine receptors, which is why it can make tiredness feel temporarily less persuasive. It does not erase the need for sleep. It simply argues with the messenger for a while, rather confidently, like a tiny lawyer in a coffee cup. During sleep, the brain also changes how it handles memory. Slow-wave sleep appears important for consolidating certain kinds of learning, especially by supporting communication between the hippocampus and wider cortical networks. The hippocampus can replay patterns related to recent experience, helping the brain stabilize useful memories and integrate them with older knowledge. Rapid eye movement sleep may support emotional memory, creative association, and aspects of learning, though the details remain an active area of research. Across a night, the brain is not merely storing everything. It is strengthening some patterns, weakening others, and reorganizing information. Sleep may also support physical housekeeping in the brain. The glymphatic system is a fluid clearance pathway involving cerebrospinal fluid movement through brain tissue. Research from Mike and Nedergaard's group has suggested that this system becomes especially active during sleep, helping clear metabolic waste products from the brain. Scientists are still refining exactly how this works in humans, but the idea is important because the brain has high energy demands and produces waste as it works. Sleep may give the brain conditions that make cleanup more efficient. Dreams remain partly mysterious. Current science links dreaming with memory, emotion, sensory simulation, brainstem activity, and cortical interpretation. Dreams can draw from recent experience, old memory, bodily feeling, fear, desire, and random fragments of thought. They are not fully understood, and there is no single accepted theory explaining every dream. What is clear is that dreaming is not simply nonsense. It reflects the sleeping brain's strange ability to generate an inner world while partly disconnected from the outer one. When sleep is disrupted or too short, the effects reach across the nervous system. Attention becomes less stable. Reaction time slows. Emotional regulation becomes harder. Learning and memory can suffer. Pain sensitivity may increase. Autonomic balance can shift, affecting heart rate, stress responses, appetite, and metabolic regulation. Even one poor night can make the world feel sharper at the edges, not because reality has changed, but because the nervous system is less well restored. Sleep, therefore, reveals something profound about the nervous system. It is not built only for receiving the world, predicting it, and acting within it. It also needs periods of altered connection, when attention loosens, sensory input fades, memories are processed, body rhythms settle, and cellular maintenance can continue. While the body rests, the nervous system is still active, but its attention turns inward. In the quiet dark of sleep, the brain is not absent. It is changing rhythm, tending itself, and preparing the waking mind to meet the world again. Sleep shows the nervous system caring for itself, restoring rhythms, processing memory, and maintaining the delicate conditions that make waking life possible. But the nervous system is not simply maintained after it appears. It has to be built. Long before a person can dream, speak, walk, remember, or wonder why sleep always feels most appealing. 5 minutes before an alarm, the nervous system begins as a developing structure, shaped by genes, cells, chemical, signals, movement, and experience. The nervous system develops through a remarkable sequence of events. Cells divide, move, specialize, extend fibers, find targets, form synapses, remove unnecessary connections, wrap some axons in myelin, and keep refining themselves through experience. It is not assembled like a machine with each part placed neatly by an outside builder. It grows from within, following inherited instructions, chemical gradients, physical constraints, and feedback from the body and environment. Development is organized, but it is not rigid. It is more like a living construction project, where the builders, materials, and blueprints are all made of cells. Early in embryonic development, a structure called the neural tube forms. The neural tube gives rise to the brain and spinal cord, which together become the central nervous system. This early formation is one of the most important steps in building the nervous system, because the basic layout of the future brain and spinal cord begins to emerge from it. Alongside this, Neural crest cells also play a vital role. These cells move through the developing embryo and contribute to many structures, including important parts of the peripheral nervous system. Some become sensory neurons, autonomic neurons, Schwann cells, and other tissues. In other words, the branching nerves that eventually reach through the body have origins in some very adventurous early cells. As development continues, immature cells become more specialized through differentiation. Some become neurons, others become glial cells. Different neurons take on different identities depending on where they are, which genes are active, and which chemical signals surround them. A neuron destined to help control movement is not the same as one that will process visual information or help regulate digestion. The nervous system needs variety, because sensing light, contracting a muscle, balancing the body, and forming a memory are not the same job. Biology, thankfully, does not try to solve every problem with one universal cell wearing different hats. Once neurons begin to form, many must send out long fibers called axons. An axon may need to grow across considerable distance to reach the right region, muscle, gland, or partner cell. At the tip of a growing axon is a structure called a growth cone. The growth cone explores the local environment, responding to chemical cues that can attract, repel, guide, or stop it. These cues help axons follow approximate routes through the developing body. The process is not random wandering. It is guided searching, with growing nerve fibers responding to molecular signposts along the way. The neuroscientist Roger Sperry helped shape understanding of how nerve fibers connect with appropriate targets through his work on chemoaffinity theory. The broad idea is that developing nerve cells and their targets carry chemical labels or cues that help guide connections. Sperry's research, including studies of visual system wiring, supported the idea that neural connections are not formed by chance alone. They are guided by biological recognition. Modern neuroscience has added many details, including specific guidance molecules, gradients, receptor systems, and activity-dependent refinement. But the central insight remains important. Developing neurons are guided toward meaningful connections. After axons reach their targets, synapses begin to form. These are the communication points where neurons influence other neurons, muscle cells, or gland cells. At first, the developing nervous system often creates more connections than it will eventually keep. This may sound inefficient, but it gives the system material to refine. Development is not only about adding, it is also about selecting, strengthening, weakening, and removing. Synaptic pruning is the process by which some connections are reduced or eliminated. It is a normal part of nervous system development, especially during childhood and adolescence. Pruning helps refine circuits so they become more efficient and useful. A developing brain does not become better by keeping every possible connection forever. It becomes better by shaping its networks around experience, activity, and biological need. This is a little like editing a long piece of writing. The final version is not improved by leaving every sentence in just because it once seemed promising at 2 a.m. Experience-dependent refinement is central to this process. Sensory experience, movement, social interaction, learning, sleep, nutrition, and health all influence how the nervous system matures. Genes provide essential instructions, but the environment helps shape how those instructions unfold. Vision, for example, depends not only on forming eyes and visual pathways, but also on visual experience during important developmental periods. Movement helps refine motor circuits. Language exposure shapes language networks. Social interaction helps the developing brain learn emotional cues, attention, communication, and regulation. Myelination is another long developmental process. Myelin is the fatty insulating layer produced by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. It helps nerve signals travel faster and more efficiently. Myelination begins before birth in some regions and continues through childhood, adolescence, and into young adulthood in parts of the brain. This long timeline matters because faster and more coordinated communication supports developing movement, attention, planning, emotional regulation, and complex thinking. The nervous system therefore develops over a very long arc. It begins before birth, grows rapidly through early life, refines itself through childhood, reorganizes during adolescence, and continues changing in adulthood. Even after the major developmental stages are complete, the brain remains plastic. It can still learn, adapt, form memories, strengthen skills, and adjust behavior. But early development is especially powerful because it lays down the basic architecture that later experience continues to shape. Genes and environment are not rivals in this story. They work together. Inherited instructions guide cell identity, growth patterns, molecular cues, and basic organization. Cellular self-organization allows tissues to form complex structures. Sensory experience, movement, nutrition, sleep, learning, and social connection then help refine the system. The nervous system is built by biology, but it is also sculpted by life. From the neural tube to neural crest cells, from growth cones to synapses, from pruning to myelination, the developing nervous system shows that the body's communication network is not simply switched on, fully formed. It grows, tests, strengthens, removes, insulates, and adapts. Every adult thought, movement, memory, and feeling rests on this long history of construction, a quiet biological unfolding that began before awareness and continues in subtler ways for as long as the nervous system keeps learning. The nervous system builds itself through growth, guidance, refinement, and experience. But even after that long development, it remains fragile in particular ways. Nerve tissue is living, specialized, and extremely organized. Its cells may stretch across long distances, connect to precise targets, and depend on carefully balanced chemistry, blood supply, insulation, and timing. When that organization is disrupted, the effects can be serious, because the nervous system is not simply a collection of parts that can always be replaced. A neuron can be a remarkably long cell. Some motor neurons send axons from the spinal cord down towards muscles in the legs and feet. Sensory neurons can carry information from distant parts of the body back towards the spinal cord. These long projections make communication possible, but they also create vulnerability. If an axon is damaged, the cell has to maintain itself, repair what it can, and sometimes try to regrow across a long distance. That is not easy. It is less like replacing a short wire, and more like trying to repair a living road that also has to find its exact destination again. The specific connections matter too. A neuron does not simply connect anywhere and produce the same result. Nervous system function depends on pathways, timing, and target relationships. Sensory information must reach appropriate circuits. Motor signals must reach the right muscles. Brain regions must communicate in organized patterns. If damage interrupts a pathway, the result depends on which fibers were affected, how badly they were damaged, and whether other circuits can compensate. A tiny injury in one place may produce noticeable effects, while a larger injury elsewhere may produce a different pattern entirely. Repair differs between the peripheral nervous system and the central nervous system. In the peripheral nervous system, damaged axons can sometimes regrow, especially if the nerve cell body remains alive and the supporting structures are still present. Schwann cells are important here. They help form myelin around peripheral axons, But after injury, they can also support cleanup and create guidance pathways that help regrowing axons move towards their targets. This does not guarantee full recovery, because distance, scar tissue, injury severity, timing, and target accuracy all matter. Still, peripheral nerves often have more capacity for regeneration than the brain and spinal cord. In the central nervous system, repair is much more restricted. The brain and spinal cord contain inhibitory molecules, scar-forming responses, complex local environments, and highly specialized circuits that make long-distance regrowth difficult. Oligodendrocytes form myelin in the central nervous system, but they do not support repair in quite the same way Schwann cells often can in peripheral nerves. After major injury, central nervous system axons usually do not regrow successfully over long distances. This is one reason spinal cord injuries can have lasting effects. The biology is not unwilling out of spite, although it can feel that way. It is simply constrained by the structure and chemistry of the tissue. Even so, the nervous system can change. Neuroplasticity is the ability of the nervous system to alter structure and function in response to experience, learning, injury, and rehabilitation. Neuroplasticity can involve changes in synaptic strength, changes in cortical maps, altered activity across networks, recruitment of remaining pathways, and new strategies for movement or communication. It is one of the nervous system's most hopeful qualities, but it should not be exaggerated into the idea that the brain can simply rewire anything at will. Biology is adaptable, not magical. The neuroscientist Michael Merzenich is strongly associated with research on neuroplasticity and cortical remapping. His work helped show that sensory and motor maps in the cortex can change with experience and after altered input. This was important because older views often treated the adult brain as much more fixed. Modern neuroscience shows that change remains possible throughout life, but also that the degree and type of change depend on age, injury, practice, attention, motivation, health, and the pathways still available. Stroke is one of the clearest examples of sudden nervous system disruption. A stroke happens when blood supply to part of the brain is interrupted or when bleeding damages brain tissue. In an ischemic stroke, a blockage reduces or stops blood flow, depriving neurons of oxygen and glucose. In a hemorrhagic stroke, a blood vessel ruptures and bleeding damages tissue directly, while also disturbing pressure and blood supply. Neurons are highly energy dependent, and without oxygen and fuel, they can lose the ability to maintain ion gradients, signaling fails, and cells may be injured or die. The effects depend on which brain region is affected and how quickly treatment occurs. Traumatic brain injury is another form of disruption. A blow, rapid acceleration, or penetrating injury can damage neurons, axons, blood vessels, and glial cells. Sometimes damage is focal, concentrated in one region. Sometimes it is diffuse, affecting connections across wider networks. Spinal cord injury can interrupt sensory and motor pathways between the brain and body. Nerve compression, such as pressure on a peripheral nerve, can disturb signaling and cause weakness, numbness, tingling, or pain. Inflammation can alter the chemical environment around nerves, making them more sensitive or less reliable. Demyelination disrupts communication in another way. Myelin helps signals travel quickly and efficiently along axons. When myelin is damaged, signals may slow, distort, or fail. Multiple sclerosis is one example of a condition involving immune-mediated damage to myelin in the central nervous system. The symptoms can vary widely because different pathways may be affected. This shows again that nervous system problems are often problems of communication, not simply problems of one isolated part. Rehabilitation works with the nervous system's remaining abilities. Physiotherapy can support movement. balance, strength, coordination, and functional recovery. Occupational therapy helps people adapt daily activities, regain practical skills, and use strategies or tools that support independence. Speech and language therapy can help with communication, swallowing, and cognitive linguistic difficulties. Neurorehabilitation brings these approaches together, using repeated practice, feedback, task-specific training, and supportive care to help the nervous system and body adapt after injury. Recovery is rarely just one thing. It may involve tissue healing, reduced inflammation, strengthened remaining pathways, new compensatory strategies, better coordination, emotional adjustment, and repeated learning. Sometimes recovery is substantial, sometimes it is partial, sometimes adaptation matters more than restoration. The nervous system can change throughout life, but not without limits. Its capacity for repair is real, and so are the biological constraints. Injury reveals both the vulnerability and resilience of the nervous system. Long axons, precise connections, myelin, blood supply, synapses, and cellular support all have to work together for communication to remain smooth. When that communication is disturbed, the effects can reach into movement, sensation, speech, memory, attention, pain, and emotion. Yet the same nervous system that can be damaged can also learn, compensate, reorganize, and slowly rebuild function where biology allows. It is not endlessly repairable, but it is not helpless either. It carries within it a quiet capacity for adaptation, shaped by cells, practice, care, and time. Injury shows how nervous system communication can be interrupted suddenly, but disease can reveal the same truth in slower, subtler, and sometimes stranger ways. Disorders of the nervous system show how normal function depends on delicate timing, chemical balance, blood supply, immune regulation, structural integrity, and the coordination of vast networks. When one part of that balance changes, the effects can appear in movement, memory, sensation, mood, attention, pain, sleep, or consciousness itself. Parkinson's disease is one clear example of how movement depends on chemical signaling and circuit control. It is named after James Parkinson, who described the condition in the early 19th century. In Parkinson's disease, dopamine-producing neurons in a region called the substantia nigra are gradually lost. These neurons help regulate circuits in the basal ganglia, which are involved in movement selection, initiation, habit, and suppression. When dopamine signaling is disrupted, movement can become slower and more difficult to start. Tremor, stiffness, changes in posture, and problems with balance may also appear. This shows that movement is not simply a matter of muscles being strong enough. The muscles may still be capable of contraction, but the nervous system's ability to select and release movement is altered. One treatment concept in Parkinson's disease is levodopa, a chemical that the brain can convert into dopamine. It can help improve symptoms for many people, although it is not a cure and long-term treatment can become complicated. Deep brain stimulation may also be used in some cases, using carefully placed electrodes to influence abnormal activity in motor circuits. It sounds slightly futuristic, but it is really a precise attempt to calm a misfiring conversation inside the brain. Alzheimer's disease reveals a different kind of disruption, named after Alois Alzheimer, who described characteristic brain changes in the early 20th century. It is strongly associated with memory decline, changes in thinking, and broader effects on behavior and daily function. Early memory problems are linked partly to damage involving the hippocampus and connected regions, though the disease eventually affects wider cortical networks. Alzheimer's disease is associated with amyloid plaques and tau tangles, abnormal protein-related changes found in the brain. These changes are part of a much larger disease process involving synapse loss, inflammation, cell stress, and network breakdown. Treating neurodegenerative disease is difficult because neurons, synapses, and networks may be changing for many years before symptoms become obvious. Once large numbers of connections are lost, restoring the original pattern is extremely hard. Modern treatments may aim to reduce symptoms, support function, slow aspects of disease progression in selected cases, and improve quality of life. Cognitive support, structured routines, caregiver education, and research into disease-modifying therapies all matter because the disease affects not only cells, but the person's whole relationship with memory, independence, and daily life. Epilepsy shows how the nervous system can be disrupted electrically. The International League Against Epilepsy defines epilepsy in relation to an enduring tendency to have seizures. A seizure is not a mysterious personal failing, and it is not a dramatic stereotype. It is a circuit event involving abnormal hypersynchronous neural activity. In other words, groups of neurons fire in an unusually coordinated and excessive way. Depending on where this happens and how it spreads, seizures can affect awareness, movement, sensation, emotion, memory, or behavior. Some seizures involve brief lapses in awareness. Others involve unusual sensations, repeated movements, confusion, or convulsions. The variety exists because the brain is organized into many circuits, and seizures reflect which networks are involved. Treatments may include anti-seizure medications, lifestyle measures such as sleep regularity, surgery in carefully selected cases, implanted stimulation devices, or dietary therapy for some forms of epilepsy. The key idea is that epilepsy is a disorder of brain excitability and network control, not a sign of weakness or character. Migraine is another neurological disorder, and it is much more than a bad headache. Migraine involves altered sensory processing, trigeminal pathways, blood vessel signaling, brainstem regulation, and changes in brain excitability. Some migraines include aura, which can involve visual disturbances, tingling, speech difficulty, or other temporary neurological symptoms. One important mechanism linked with aura is cortical spreading depression, a slow wave of altered electrical and chemical activity moving across the cortex. Migraine can bring sensitivity to light, sound, smell, movement, nausea, pain, fatigue, and cognitive fog. It shows how the nervous system's sensory filters can become intensely sensitive. Modern migraine treatments may include acute medications, preventive medications, lifestyle management, and newer therapies that target calcitonin gene-related peptide pathways. The existence of these treatments reflects a more precise understanding of migraine as a nervous system disorder, not simply a person being bad at tolerating discomfort. Multiple sclerosis reveals the importance of myelin and glial support. Jean-Martin Charcot helped describe multiple sclerosis as a distinct neurological disease in the 19th century. It is an immune-mediated demyelinating disease of the central nervous system, meaning the immune system attacks myelin in the brain and spinal cord. When myelin is damaged, signals may slow, distort, or fail. Symptoms can vary widely because different pathways may be affected, including those involved in vision, sensation, movement, balance, fatigue, and coordination. Treatments for multiple sclerosis include medications that aim to reduce immune activity and lower the risk of relapses or new lesions, along with rehabilitation, symptom management, exercise where appropriate, and support for daily function. It is a powerful reminder that nervous system communication depends not only on neurons, but also on insulation, immune balance, glial cells, and the chemical environment around axons. These disorders differ greatly, yet they all reveal something about the nervous system's normal work. Parkinson's disease shows the importance of dopamine and basal ganglia circuits. Alzheimer's disease reveals the fragility of memory networks and synapses. Epilepsy shows the need for controlled electrical timing. Migraine reveals the sensitivity of sensory and vascular signaling. Multiple sclerosis shows how essential myelin is for reliable communication. The nervous system can be disrupted gradually, suddenly, electrically, chemically, structurally, or immunologically. But studying these disruptions has also led to treatments, support strategies, and deeper understanding. Medications, deep brain stimulation, rehabilitation, cognitive support, lifestyle management, and disease-modifying therapies all grow from the same basic principle. If the nervous system is a system of communication, then treatment often means trying to restore, protect, guide, or compensate for that communication. In illness, the nervous system reveals its vulnerabilities but also the extraordinary precision that makes ordinary life possible. The nervous system is easiest to misunderstand when it's imagined as one thing doing one job. A brain thinking, a nerve carrying a signal, a reflex pulling a hand away, a memory sitting somewhere in storage. Each of these images contains a little truth, but none of them is large enough. The nervous system is not a single command chain. It is a living conversation, constantly moving between brain, spinal cord, peripheral nerves, muscles, organs, senses, immune signals, hormones, gut, skin, and environment. Every moment of ordinary life depends on that conversation. A sound enters the ear, but it only becomes experience after sensory cells, nerves, brainstem pathways, thalamic filtering, cortical interpretation, attention, and memory have all played their part. A hand moves, but only after intention, planning, spinal pathways, peripheral nerves, the neuromuscular junction, muscle fibers, and sensory feedback have turned thought into action. A heartbeat changes, digestion slows or settles, body temperature shifts, pain rises or fades, and breathing adjusts its rhythm, often without conscious instruction. The nervous system is not only the machinery of thought, it is the machinery of being bodily alive. This matters because thought is only one visible surface of nervous system activity. The same system that allows language, memory, imagination, and reflection also helps regulate posture, balance, swallowing, sleep, digestion, blood vessels, pupils, and survival responses. It receives signals from the skin and muscles, but also from the lungs, stomach, intestines, heart, and immune system. It listens to the world outside and the body inside at the same time. Sometimes it produces clear awareness. Sometimes it simply keeps the body steady while awareness drifts elsewhere, possibly wondering why walking into a room so often makes the original purpose evaporate with suspicious timing. The brain and spinal cord form the central nervous system, but they are not isolated rulers. They depend on peripheral nerves reaching through the body, sensory receptors translating physical energy into neural signals, glial cells maintaining the environment around neurons, and blood vessels delivering oxygen and glucose. They also depend on hormones, immune molecules, gut signals, and body rhythms. The nervous system is physical all the way down. Electrical impulses, chemical synapses, membranes, ions, neurotransmitters, myelin, receptors, and cellular energy all matter. Conscious experience may feel airy and private, but its support system is wonderfully biological. And yet consciousness remains one of neuroscience's deepest open questions. Scientists can study attention, perception, memory, sleep, anesthesia, brain injury, and neural activity with increasing precision. They can map which networks are active during certain tasks, which regions are necessary for particular abilities, and how sensory information becomes available for report. But there is still no single agreed explanation for why certain patterns of brain activity are accompanied by subjective experience. The mystery is real, but it is not an invitation to abandon science for vague mysticism. It is a careful boundary. There is much that neuroscience has explained, and there is still much it has not fully solved. Francis Crick and Christoph Koch helped bring consciousness into modern neuroscience by arguing that subjective experience could be studied through its neural correlates, the brain processes associated with conscious awareness. Stanislas Dehaene is closely associated with global neuronal workspace theory, which suggests that information becomes conscious when it is made broadly available across large-scale brain networks, especially those involved in attention, report, and flexible control. Giulio Tononi developed integrated information theory, which tries to describe consciousness in terms of how much information a system integrates as a whole. Anil Seth has explored consciousness through predictive processing and controlled hallucination, the idea that perception is an actively constructed model shaped by the brain's predictions and sensory signals. None of these theories has fully solved consciousness. Each has strengths, open questions, and critics. Global neuronal workspace theory connects well with attention, reportability, and brain-wide broadcasting. Integrated information theory tries to address why experience might belong to a system as an integrated whole. Predictive processing offers a powerful way to think about perception as the brain's best controlled guess about the causes of sensory input. Together, these approaches show real progress, not final certainty. The science has moved far beyond simple speculation, but it has not reached a neat ending tied with a ribbon and filed under "problem solved." Researchers also made progress by studying altered states. Anesthesia can reduce or disrupt conscious awareness in controlled medical settings, offering clues about the brain activity required for experience. Sleep shows how awareness can fade, change, dream, and return. Brain injury can reveal how attention, memory, language, movement, and self-related processing depend on particular networks. Perception research shows that the brain does not passively record the world, but builds a usable model from incoming signals, prior expectations, attention, and body state. That brings the whole nervous system back to an ordinary quiet moment. Sitting still in a room may seem simple, but it is built from countless hidden processes. The brain is filtering sound. The skin is sensing pressure. The spinal cord is carrying traffic. Peripheral nerves are reaching outward and inward. Glial cells are supporting neurons. Blood flow is sustaining tissue. The autonomic nervous system is regulating heart rate, digestion, and temperature. Memory gives meaning to what is familiar. Attention decides what matters. Prediction helps the world feel stable rather than chaotic. Even the feeling of being a single self is supported by many layers. The insula helps represent internal body state. The prefrontal cortex helps guide attention and interpretation. The hippocampus links experience with memory and context. The brainstem supports arousal and basic life rhythms. The body sends constant information upward. The environment presses in through light, sound, temperature, and touch. The self is not floating above biology. It is made possible by biology, moment by moment. So the nervous system deserves to be explained slowly because it is not one mechanism with one purpose. It is communication, regulation, prediction, protection, learning, repair, and adaptation woven together. It lets the body notice the world, but it also lets the world become experience. It lets muscles move, organs adjust, memories form, emotions rise, sleep reshape the brain, and attention settle on one small part of a much larger signal stream. The nervous system is both intimate and hidden. It is closer than anything because every sensation, thought, movement, and feeling depends on it. And yet, it works mostly out of sight, in darkness, through tiny signals crossing tiny gaps. Beneath awareness, it keeps speaking in electricity and chemistry, in rhythm and silence, in action and restraint. Through that living conversation, the body does not merely exist in the world, it feels the world, responds to it, remembers it, and quietly becomes aware of being here. The nervous system, explained slowly, is not a single thread of thought running through the body. It is the body's constantly active communication system, turning signals into sensation, movement, memory, regulation, emotion, sleep, and awareness. It is slow to understand only because it is so layered. In the body itself, it is never truly slow. It is always listening, adjusting, filtering, predicting, and responding. It begins with neurons, the living messengers that carry electrical impulses through membranes shaped by ions, voltage, and timing. It depends on action potentials traveling along axons, synapses passing signals from one cell to another, and neurotransmitters such as glutamate, gamma-aminobutyric acid, acetylcholine, dopamine, serotonin, norepinephrine, and endorphins helping circuits communicate. It also depends on glial cells, the quiet support system that nourishes neurons, shapes synapses, protects tissue, clears debris, and wraps axons in myelin so signals can move with speed and precision. From there, the brain and spinal cord form the central pathways of control and interpretation, while peripheral nerves reach into the skin, muscles, joints, organs, glands, and blood vessels. Sensation enters through specialized receptors. Movement begins as intention and becomes action through motor circuits, spinal pathways, peripheral nerves, and the neuromuscular junction. The autonomic nervous system keeps the inner body regulated, guiding heart rate, digestion, breathing patterns, temperature, blood flow, and pupil size while awareness is busy elsewhere. Emotion shows that feeling is not only in the mind but in the whole body. Memory shows that the nervous system is changed by experience. Attention and prediction show that the brain does not simply record the world, but builds a usable version of it from signals, expectation, and context. Sleep shows that rest is active, with changing brain rhythms, memory consolidation, sensory disconnection, and neural housekeeping. Development reveals how the nervous system builds itself through growth, guidance, pruning, myelination, and experience. Injury and disease reveal its fragility, but also its capacity for plasticity, adaptation, treatment, and repair where biology allows. Modern neuroscience has explained an extraordinary amount about signaling, circuits, development, plasticity, sleep, pain, and disease. And still, consciousness remains partly unresolved. The quiet mystery of how physical activity in living tissue becomes subjective experience. So even now, as the body rests, the nervous system continues its hidden work. It keeps the breath moving, the heart adjusting, the skin sensing, the muscles settling, and the mind gently open to the world